I'm going to switch gears a little bit, and we're going to move on to talking about a new project that I have that we're working on in the lab that is genetically engineering macrophages, and we're using that to overcome the microenvironment in brain tumors. Now, as I'm sure I don't have to tell anybody in the audience here, childhood brain tumors are absolutely devastating for patients and their families. They remain the most common solid tumor in children, with about 19% of childhood cancers are brain tumors, meaning that we're going to have about 4,600 cases expected to be diagnosed in 2015. In general, treatments include chemotherapy and radiation, and occasionally surgery. And of course, chemotherapy and radiation affect primarily the dividing cells, which in children who are rapidly growing are going to have some devastating side effects. These can include neuroendocrine dysfunction, cognitive defects, and long-term risk of secondary malignancies. And so, of course, we're all sold on immunotherapy here, so helping these children by developing immunotherapies for brain tumors is going to be a huge step forward for their quality of life. However, this is, of course, challenging in solid tumors because of the tumor microenvironment, as some of the other presenters in this session have already touched on. Just so we're all on the same page, I'd like to introduce briefly some of the crosstalk that occurs between tumor and immune cells within the microenvironment. So it's not simply just cancer cells existing within the normal cells. We also have fibroblasts that are present. Those are going to be cranking out growth factors and generally supporting the growth and development of the cancer cells. The growing tumor then is also going to recruit myeloid cells, both from the circulation as well as from the surrounding tissue, and immediately get those to become a more anti-inflammatory, pro-tumor type of phenotype, these M2 cells that Amy was just talking about. And then in addition, we're also going to have the presence of regulatory immune cells, like our FOXP3 positive Tregs, whose job it is to basically shut down any anti-tumor immune responses. And what this all means is that when we do have cytotoxic immune cells, be they the patient's own unmodified cells or engineered cells like CAR T cells, they're going to have a really uphill battle to both localize to and then be effective within the tumor microenvironment against these tumor cells. And of course, as scientists, we also have a major uphill battle because of the number of ways, the number of mechanisms by which this tumor is shutting down the immune response. And that includes by any number of cells in the tumor microenvironment, the production of anti-inflammatory cytokines, expression of inhibitory ligands, such as the ligands for PD-1. We're going to have reduced antigen presentation by the myeloid cells. And in general, there's just going to be an induction of a Th2 type response upon arrival of T cells within the microenvironment. And just as an example of what this actually looks like, not in a cartoon, I'd like to present some of the data from my lab looking at adult glioma and the grade 4 glioma, which is glioblastoma. And what you can see is that in normal and grade 1 and 2 have a slight infiltration of these CD163 positive macrophages, which are these anti-inflammatory pro-tumor type macrophages that we saw in the previous talk. In grade 3, we definitely see an increase, and even more dramatically in grade 4 gliomas or glioblastomas. And this is very clearly illustrated by this quantification here. Now, these CD163 positive macrophages are going to be pumping out factors that are going to be shutting down the immune response within the tumor. Expanding on this a little bit, I'd like to switch to focusing a little bit more on some pediatric brain tumor data. Then, unfortunately, there's just not a lot out there published about the pediatric brain tumor microenvironment. But I've read some papers by the Foreman Lab at the University of Colorado. And what I'd like to show here is that there's some differential immune effects within the tumor microenvironment. And so we have pilocytic astrocytoma and ependymoma with greater infiltration of both CD4 and CD8 positive T cells than in GBM or medulloblastoma. This also holds true when you're looking at the total myeloid cell compartment. However, when we break it down to whether or not these myeloid cells are acting more immunosuppressive or whether or not they have the more M1 type of phenotype, what you can see is that GBM and medulloblastoma myeloid cells tend to have much higher expression of CD163 and CD206, meaning that they are much more immunosuppressive. And so at this point, I think it's pretty clear that there's a great need for some sort of product that's going to transform the tumor microenvironment to one that supports immune-mediated cancer cell destruction. And so with that, I'd actually like to introduce the topic of my presentation today, and that is my lab's creation of a novel way to treat solid tumors, including brain tumors, which is the creation of genetically engineered macrophages. Now, I'm often asked, why macrophages instead of some other cell type that is maybe a little bit easier to work with? And part of that is that monocytes, which are macrophage precursors, are currently a waste product during the production of CAR T cells by groups like Seattle Children's Therapeutic Cell Production Corps, who create the CAR T cells for the patients in our clinical trials. And so if we're able to repurpose this waste product without needing to draw any additional blood from our patients, that's great. That's a win-win for everybody involved. It also helps that we're able to capitalize on what tumors do naturally, that is recruit and retain myeloid cells. So these macrophages should be able to stay within the tumor microenvironment. And when they're there, we expect that they're going to be a great source, great local source for CAR T cell support. We also have the advantage of flexibility when it comes to macrophages. We've been able to show that there are multiple ways that you can engineer these macrophages, and importantly, you can introduce multiple transgenes, which just means that we're going to be able to take many different approaches here. And finally, thinking about the role of macrophages both within the normal immune system and in normal tissues as well as in cancer, macrophages really are the ones that are kind of directing the crosstalk between several different cell types. And so if we're able to repurpose those cells and direct them to do something different, we can really redefine the entire conversation between the tumor microenvironment resident cells, including the tumor and the cytotoxic immune cells. However, engineering macrophages is not without its technical challenges. And part of that is that, evolutionarily, they are designed to be refractory and resistant to infection by any viruses or any other sort of manipulations. And in particular, lentivirus, which works extremely well to transduce T cells and many other cell types, lentivirus cannot actually infect myeloid cells naturally. And that's because of the expression in myeloid cells by this factor called SAMHD1, which directly inhibits the viral reverse transcriptase, which means that you can't even unpackage the virus and get it into the genome and express their transgenes. However, we are deeply indebted to Dr. Nathaniel Landau's lab at NYU, who has developed a packaging system with a protein called VPX that inactivates SAMHD1. And they're HIV biologists, so they understand this a lot better than I do. But basically what happens is when you package virus with this VPX, the VPX inactivates SAMHD1 within the myeloid cell once the virus has entered, induces its degradation, and the genome of the virus can then be transcribed and imported into the nucleus. And because of that, we're able to use lentivirus with this VPX system to engineer our macrophages. And in terms of the production of lentivirus itself, it ends up looking very much the same as what we're all familiar with here, where we have a co-transfection of the entire cocktail of the accessory, or of the different viral factors. We have our transgene on an HIV backbone, and then the only addition is this VPX here. We transfect that into 293 T cells. The virus is packaged and secreted into the supernatant, and we then purify it. And at that point, we can use it to infect the CD14 positive monocytes that we isolate from PBMCs. And concurrent with that infection, we use GM-CSF to polarize these monocytes during their differentiation to a pro-inflammatory macrophage phenotype. And you can see the effect of this VPX here when I show you what these monocytes look like after infection. So this is 14 days after their infection with a virus with a lentivirus encoding GFP. You can see that the vast majority of these cells are HLA-DR or MHC class II positive, which suggests that this differentiation in GM-CSF has been persistent because these are still maintaining this pro-inflammatory type of phenotype. And when we don't use any virus, or we use virus that has not been packaged with VPX, even at an MOI of about 10, we don't see really any GFP positive cells at all, maybe 1%. And contrast that to when we use virus with VPX, we can get essentially 100% of these macrophages expressing transgene, which I think is pretty exciting. And that's recapitulated when we look at another myeloid cell marker, CD11B. Again, we have essentially 100% GFP production in conjunction with the markers that we would expect from the phenotype we've used to differentiate. And I'd like to just quickly point out here that this is 14 days post-infection. We have data out to 28 days post-infection, and we suspect, and it looks essentially the same as this, and we suspect that these phenotypes are extremely stable over the long term, which is great, and both for what we're hoping to do with patients, and also we can see this in mice. We've shown that our macrophages persist in mice, and the model that we've been using is an NSG mouse with a U87 tumor engrafted through an intracranial injection, and we're using this model in conjunction with Dr. Jensen's group, and our hope is that we're going to be able to find really nice ways that we get our genetically engineered macrophages interacting and synergizing with his CAR T cells, which we think is going to be a really powerful approach for these solid tumors. And so what we can see is that after about, we inject these gems after 7 days, and we already have tumor coming up, and that persists through the another 3 weeks through to our experimental endpoint, which you can pretty clearly see here. Now, I don't have data right at this moment showing if these gems directly affect our tumors, but we actually have that experiment underway just down the street. So unfortunately, no data for that today. And then just in my last couple of minutes here, I'll just give you a little snapshot of some of what we're looking at in terms of the constructs that we want these gems to express. Now, as Amy was mentioning earlier, these M2 macrophages in the tumor microenvironment are a huge problem. And if we're able to get those kind of reactivated, polarize them towards a more pro-inflammatory anti-tumor phenotype, that's going to give us hopefully the response that we're looking for. So what we want to do is engineer gems that are able to drive TLR4 signaling. And obviously, we can't just give LPS. We can't get these gems secreting LPS. So instead, we're taking a different approach. We're looking at expressing proteins that are known to be damage-associated molecular patterns, or DAMPs. For example, a protein called HMGB, which is secreted from dead cells. And I have made a construct that looks just at the B box of HMGB, which contains the TLR4 binding domain. And we can see in green here that this B box is nicely secreted from mammalian cells, and that when we add conditioned media containing this B box to M2 polarized macrophages, we can see just a little bump, just a little nudge in the secretion of these pro-inflammatory type of cytokines by the tumor-resident type macrophages. And we're hoping that even just this little nudge might be enough to direct changes within the tumor microenvironment that help to eliminate the tumor. And on the flip side, we also want to make sure that the gems aren't co-opted by the tumor themselves. And to that end, we are using the CRISPR system, which has already been explained, so I'm not going to go through it in any sort of detail, to knock out expression of, for example, TGF-beta or IL-10 or some of the ligands for PD-1. And what you can see is that when we use lentivirus to express this system in macrophages, we do get about a 50% decrease in the amount of IL-10 that is being produced by these cells. And we also, by qPCR, can see, again, about a 50% reduction of IL-10 or TGF when the macrophages have been transduced with the guide RNAs and Cas9 for those specific genes. And I'd like to point out that we have yet to optimize this, and we suspect that we're going to be able to get a much stronger knockdown of these genes after we optimize. And so then in conclusion, I'd just like to mention that this is just the very beginnings of this project, and it's really exciting to work on something that is, we think, so very flexible that we can potentially disrupt resistance to the TME. We can, look at anti-tumor immunity. We can hopefully help the immune localization of T and NK cells, and potentially down the road, even look directly at tumor inhibition. And since this is a new project and there's so many people that are in this room that are very, very smart, if anybody has any really creative ideas, I'm really looking forward to hearing them. So feel free to find me afterwards. And with that, I would like to just thank all of the members of the Crane Lab, past and present, who have helped get this project off the ground. In particular, Kara, another postdoc in the lab with me, has been working on this. And also several members of the Jensen Lab, in particular Michael Baldwin and Cindy Chang, who have been very, very helpful with getting our mouse project off the ground. And then also deserving of special mention is one of our funding agencies, the Stephen Higgins Brain Tumor Research Fund, who has really believed in these gems right from the outset. And with that, I would be happy to take any questions within this panel discussion we're about to have. Thank you.